Hello, and thank you for joining us. I am Jasmine Navarez, Digital Content Coordinator at Simulations Plus. I'll be hosting today's webinar behind the scenes, making sure we don't have any technical difficulties. In this webinar, senior scientist Revathi Chapa and scientist 2 Suvarchala Avvari will demonstrate how standard DDI models have been validated in GastroPlus, as well as how the software can be used to characterize and validate the effect of transporters on drug absorption, systemic distribution, and systemic clearance. Following the presentation, our presenters will be available for a live Q&A. A few housekeeping items before we begin. We take our privacy rights seriously. By registering for and attending this event or participating in the Q&A, you're allowing us to contact you for follow-up. You may ask questions via the questions panel on your dashboard at any time. If you need assistance, please use the hand raise icon. We'll address all questions in the Q&A after the presentation. Before we begin, I'd like to conduct a short poll to better understand your familiarity with the DDI predictions. And I'll go ahead and launch that now. And I'll give everybody a couple seconds here to answer the first poll. Okay, is the answers coming in? All right, and I'll go ahead and end the poll here. And I'll pass it over to Suvarchala to start the presentation.
Thanks, Jasmine. Yes.
Is Revathi Chapa. I'm working as a senior scientist at Simulations Plus, essentially working on various PBPK applications, be it drug-drug interactions or the first in human. So today I'm here along with my co-presenter Suvarchala. So we both will be presenting on development and application of PBPK models to support transporter-mediated DDIs. So Suvarchala, if you can give a brief intro of yourself.
Sure. Thanks, Revathi. So, hello everyone. My name is Suvarchala Avvari. I'm scientist 2 at Simulations Plus. So, I joined Simulations Plus in 2021 September, and I've been working on various applications like first in human predictions, formulation assessments, drug-drug interactions, and also testing of various versions of GastroPlus. So, yeah, thanks, Revathi. So, floor is yours. You can start with the presentation.
Sure. So yeah, the outline of today's presentation will be, we will be discussing the types of drug-drug interactions. Then I would give you a brief overview of how the DDI module interface looks in GastroPlus. And then we will also have overview of relevant regulatory guidance documents where we will be discussing the investigation of transporter involvement in drug clearance or the investigation of transporter inhibitory potential. And then we would be discussing some PBPK models, especially PGP substrates, which are digoxin and fexofenadine, and OATP1B substrates, which includes rosuvastatin and pravastatin. And also we would discuss how we approach the predictive performance in terms of if we have any in vitro or in silico data and how we make some prospective predictions for regulatory submissions. Okay, so coming to types of interactions within GastroPlus, you can handle two types of interactions. One be a steady state and other is a dynamic simulations. So in steady state where we are kind of approximating a situation where all the processes are at a standstill. So it has reached some steady state. Whereas in dynamic, all the processes are continuously changing. When I mean all the processes here, we mean the absorption, distribution, metabolism, eliminations, and also the pharmacodynamics. So this is a continuous process and we are looking at what is happening at any given time. Coming to types of interaction, we are trying to understand competitive, time-dependent inhibitions and inductions. In competitive inhibition, it's necessarily a competition between the compound and the perpetrator. Here, it could be an inhibitor, which is kind of competing for the same site of enzyme or transporter. So that's essentially the competitive. Whereas in time-dependent inhibition, some compounds cause an irreversible effect on the enzyme or transporter, and this could lead to the ultimate degradation of that enzyme or transporter, which eventually needs some resynthesis to come back to regain its normal activity. Whereas in induction, there if there is any some kind of stimulus, there could be some synthesis of the a new enzyme or transporter that could be happening based on the stimulus. So this is essentially the induction. So GastroPlus has these capabilities to inculcate or incorporate these kind of inhibitions if your compounds are interacting through any of these mechanisms. So coming to the dynamic simulations, here you can see we have different equations for competitive and time dependent and also induction. So here it's not just the effect of one compound. You can try to simulate the effect of multiple substrates, multiple perpetrators on one substrate, or you can handle multiple substrates with one perpetrator. So it could be either ways. So again, these are the set of equations that we employ for competitive inhibition, time-dependent inhibition, and induction. You can get into these equations these are well described in the GastroPlus manual. Okay, so before we do any kind of DDI predictions, it is always an imperative to have a right PBPK model, which is always linked to a cat physiological intestinal model. By this, I mean that it is always important and necessary to have a right processes defined when we wanted to make the right approximate DDI predictions. For example, when a drug is entering into the body, in the lumen, it could undergo several processes. For example, first the drug needs to be dissolved. Some drugs could precipitate based upon the pH, and then the drug could also potentially degrade if the pH is not comfortable for the drug. And then the drug gets into the solution, then it needs to traverse through the gut wall. So when it traverses through the gut wall, it has to overcome two barriers where it could be either effluxed by the efflux transporters, or it could be metabolized by the enzymes present in the gut wall. So it has to overcome these two processes in order to make it to the systemic circulation. So there could be sometimes there could be transporters which could be facilitating the absorption of these drugs, which are essentially the uptake transporter. So before we make any DDI predictions, it is very important to have a good understanding on processes drug could go through before it makes up to the systemic circulations. So these processes are happening simultaneously in all of the compartments. When we say all of the compartments, we refer to the jejunum, ileum, duodenum, and colon, and rectum. Okay, so as I said, before we make any DDI prediction, it is a prerequisite to have the right dissolution and absorption. Otherwise, you would be missing out on some processes which are sensitive for predicting the DDI. So it is essential to incorporate the metabolism and transporters if they are involved in the absorption processes. Okay, so here is snapshot of how we develop our models, which involves developing a DDI qualification matrix. So these qualification matrix includes a different range of PBPK models for probe substrates, inhibitors, inducers. So remember, these models are in various stages of validation with the information coming up and evolving over the time. So we try to incorporate the new mechanisms also, and we do refine the models periodically so that it reflects the current understanding of drug DMPK. So here is a comprehensive list of models that we have in-house and are frequently worked on and validated with the new information. But this is not just the list. We are also working continuously on developing new models based on the regulatory interest. And apart from that, any sponsor or innovator can also work on their compounds and create PBPK models if they think that their compound can have some inhibitory potential for any transporters or enzymes. Okay, so now I would be giving you a brief overview of the DDI module interface in GastroPlus. Okay, so this is how the basic interface layout looks like. You see, there's a partition in between where on one side you could either select your victim or perpetrator, and on your right hand side you could select your alternate compound. For example, if you select here the victim, here you could select your perpetrator. So as you can see, the prediction types, there are two types. One is steady state prediction and another is dynamic simulation. So GastroPlus do offer both types of predictions. Remember that steady state predictions are usually confined to the early phase of drug development when innovator is trying to get a sense of what could be the prospective possible DDI, but eventually when it comes to regulatory submissions, dynamic simulations are more desirable because it reflects all the processes that are important for a drug when it is traversing throughout the body. So this is the left hand side. We ideally prefer to put the victim on the left hand side of the compound so that since the GastroPlus picks the physiology, the PBPK physiology and gut physiology from the left hand, whatever the compound you have open, so it is always desirable to put the victim compound on the left side. And then alternatively, you can put your perpetrator on the right hand side. So again, if you look at the simulation mode, we do offer quite a range of simulation modes. For example, single simulation, which could be a representative of a mean simulation of the population, and then could be PopSim, where there is some variability coming because of certain reasons, because of the polymorphisms in enzymes or transporters or because of any other dissolution related variability. These are the two types of mode that would be of interest, especially with respect to DDIs. So, okay, so again, this is the right hand side where it's desirable to place your interacting compound. The interacting compound is your perpetrator, which could be your inhibitor or an inducer. And when I say inhibitor, it could also be competitive inhibitor or a time dependent inhibitor. So you could specify this your perpetrator here. So here is just an example where we opened a project we created for rifampicin where it is vigorously validated with various CYP3A4 substrates or ATP substrates, BCRP and PGP substrates. So this is a well validated project that we created for rifampicin, and this is one of the drug file where we studied this study from Procuracerent from 2014 paper where they have administered rosuvastatin with rifampicin, and we are seeing how the effect of rifampicin on rosuvastatin just for a demonstration. So here also you can see on your right hand side, whenever you are selecting your either the prediction type, the steady state or dynamic, you could have this FM for steady state predictions. It's essentially coming from the data you present either as a Vmax or Km or through the intrinsic clearance. So this is how it is presented here. So it is important to define your metabolisms and transporter pathways rightly to get the right DDI predictions because sometimes there could be overestimation or underestimation if one of the pathways are given a higher or lower contributions. So this is how the FM table looks like. And then you can also get a visual representation of how your metabolic profile in gut and liver is displayed in the pie chart. And then, for example, you can define your simulation length. For example, in this case, rifampicin is given for 30 minutes as an IV infusion, and then rosuvastatin is administered after half an hour. So the simulation length was 24 hours, or in total, the simulation length would be 24.5 hours. So you could define the simulation length here. So the way you would define the physiology administration of your substrate or victim compound is you can open a mixed multiple dose information in the drug file, and you can define your drug administration. For example, I have said that the rifampicin is given for 30 minutes and then the compound rosuvastatin administered. So it is administered at 0.5 hours. So these are the physiologies that we define based on whether it is given in fasted condition or fed condition and how long it takes to come back to the fasted condition. So this is important because this compound has intrahepatic circulation and it kind of contributes for the reabsorption of the drug. So this is how we specify substrate/victim settings. Okay, so coming to the perpetrator settings, for example, rifampicin is a pan inhibitor of enzymes and transporters. So it has different inhibition potential, different inhibitory potentials for different enzymes and transporter. For example, the Ki that we optimized for OATP 1B1 and 1B3 is around 0.07 micromolar, whereas for BCRP it is around 14.9 micromolar. So these represents the parameters that are previously verified with relevant substrate. For example, we verified these interactions with OATP1B substrates like rosuvastatin, pravastatin, and atorvastatin. And for BCRP, we did based on rosuvastatin as an example. So similarly, we do have interactions for other enzymes and transporters. So whenever a sponsor is working with a new model. So we do offer this rifampicin project where you could select the relevant inhibitory potentials. For example, you could highlight them. I mean, they are usually highlighted and validated as true whenever the parameter is validated to make sure that if your compound is a OATP1B substrate and you think that what could be the DDI with the rifampicin, you can use these inhibitory potentials to get a sense of how much rifampicin could impact the AUC or the Cmax of your compound. So this is just an example to demonstrate. Okay, and then you could also give the dosing information of the perpetrator here, or you can also alternatively have an MDD file in your perpetrator compound that defines the dosing information, like the way that I showed for substrate. So we can do that for perpetrator as well. So once you select all the relevant interaction parameters, here I wasn't showing what parameters I was selecting, but I did select parameters for 3A4, OATP 1B1, 1B3, BCRP, OATP 2B1. So those we need to select those interaction parameters and first run a baseline simulation. So when we say baseline simulation, here the compounds, both the victim and the perpetrator are run as if they are administered separately, means there is no kind of interaction that is happening. But when we wanted to see the DDI effect, we need to run full simulation where we could see how much your compound rifampicin is causing a change in AUC or Cmax of the victim or substrate, here rosuvastatin. So as you can see, once the simulation is finished, you would get this dynamic simulation output. For example, you can see how much of your FA is changed, or FDP, which is a fraction that makes up to the portal circulation, and your change in bioavailability. You could see how your Cmax, Tmax, and AUC, or AUC 0 to T and AUC 0 to infinity changes. So you can get the values for the baseline, the DDI, and also the GastroPlus gives you the ratios based on the DDI upon baseline. So you could see that ratios are calculated here. So, and then you can also see the plots, how they show. For example, this is on your left-hand side, a baseline simulation where your compound and the compound rosuvastatin and rifampicin are administered, but there is no interaction. But when we see a full dynamic simulation, you can see here, because we have selected certain inhibition towards OATPs, BCRPs, so we do see some kind of effect because of that. And we see that increase in the plasma concentrations because the transporters which are primarily involved in the disposition of rosuvastatin are now inhibited. And now we see an increase in plasma concentration. So we do see an increase in DDI ratios. Okay, so that's the introduction on our GastroPlus DDI module interface. But you could get complete information and you can refer to our GastroPlus manual. So, okay. So there are different types of guidance documents to aid the sponsors who are navigating through developing a new molecule. So for example, here now we are looking at a clinical DDI guidance from FDA. So for example, this is to show you how the PBPK models can be used when with the help of information that we get from in vitro as well as clinical data. For example, if your compound is substrate for a particular enzyme, like for example, take the example of CYP3A4 or 2D6 or 2C8. So, ideally, a mass balance study is done, and also a DDI with a strong enzyme inhibitor. For example, if you're referring to 3A4 at rifampicin is used to see how much it is changing the AUC of your AUC and Cmax of your compound. Based on that, then we could get initial the estimates of FM, how much your metabolism is contributing. Similarly, if there are transporters, you could also see how much of your FT, which is a fraction transported by specific transporter. And so based on that information, we do get initial sense of FM and FT. So now we can make other types of predictions. For example, now you can try to see the effect of moderate and weak inhibitors or inducers to see how much, if the drugs are co-administered, how much the possible effect could be. So you can use a PBPK modest instead of doing a clinical DDI studies, because we do have the information on FM and FTs based on a strong clinical DDI study. Okay, so these are another few set of guideline on the investigation of drug interactions. This is from EMA. And then there is also drug interaction studies, which is ICH harmonized guideline. So these guidelines are very thorough in terms giving the sponsor clues on what kind of in vitro data needs to be generated, what kind of clinical data is imperative to understand the mechanisms which are substantial for the absorption and elimination of the drug compound. So, and they also guide you how to use PBPK models where you could avoid doing some of the clinical DDI studies because with validated PBPK models. And then we also have in vitro DDI guidance from FDA. This is kind of very helpful when a sponsor is in his early phase of drug development and doesn't have much of the information related to what kind of enzymes, what kind of transporters are involved. So here is FDA's outlook. Like FDA recommends a screening for these transporters. For example, FDA wants us to screen for PGP, BCRP, OATP 1B1, 1B3, organic anion transporter 1 and 3, organic cation transporter OCT2, and mates, which are multi-drug and extrusion transporters 1, 2K. So this is the list which is more consensually accepted by all regulatories, but there is additional recommendation, especially with respect to EMA, where which involves the screening for organic cation transporter 1, OCT1, bile salt export protein, which is BSEP, and MRP2, which are involved in the hepatobiliary transport. Okay, then so here is the regulatory requirements of transporter evaluation. This is comparing how different regulatory recommendations vary based on what flex and uptake transporters could be involved for the screening process. As you can see, all the regulatory agencies agree upon PGP and BCRP, but there's no any particular recommendation from FDA or NMPA or PMDA with respect to BSEP and MRP2, whereas EMA recommends to consider for screening for these transporters. And then if the hepatic pathway or elimination is significant, that is the uptake in the liver is clinically important. The other factor needs the importance of active transport into a liver, especially by 1B1 and 1B3 needs to be screened. Again, it's the same recommendation from NMPA and for PMD and EMA. So if the pathway has to be more than 25%, because that's when you would see a significant change in AUC, that's why they recommend that it needs to be at least 25% of the elimination should be hepatic. If it needs to be screened for hepatic transporters like OATP 1B1 and 1B3. And if it is for renal transporter, again, it needs to be 25% of the elimination through renal secretion. It has to be screened for OAT1, 3, OCG2, and MAT1 and MAT2K. Okay, so FDA provides a decision-making tree to help navigate when these transporters needs to be screened and when they are clinically could be causing a clinical effect. So here, as you can see, this is a decision tree that is provided from FDA side, where all investigational drug needs to be screened for PGP and BCRP. So no matter what, we need to screen if the compound is a substrate for PGP or BCRP. So when it comes to hepatic or biliary secretion, if it is more than 25% of the total clearance, then we do need to consider if it is a substrate for OATP 1B1 or 1B3. And then if it is the renal clearance is more than 25% of total clearance, so this can be the information, the renal clearance greater than 25% can be obtained if the majority of the compound is excreted renally. So then we need to screen for this transporter. So ideally, a mass balance study kind of gives you what kind of major pathways are major elimination pathways, and it helps us to give certain direction, whether it is hepatic dispersion that is predominant or renal secretion that is predominant. So here is another guideline from EMEA where it is more or less kind of agrees with FDA. So for example, here again, we are looking at a ballpark of more than 25% elimination as hepatic. So if it is more than 25%, we need to screen for transporters like OATP 1B1 and 1B3. If it is no further studies are needed. But if it is yes, we need to do a clinical DDI study with potent inhibitor, like for example, rifampicin. So if there is a significant interaction, we need to indicate in the label how the recommendations are, if the drug, how the recommendation, if the drug needs to be co-administered, or what kind of label changes are essential to see how it reflects in the DDI, how it affects the PK of this compound. So, and if it's no, then there are no further studies that is required. Similarly, for renal elimination, it has to be more than 25%, and then we would screen for the transporters involved. We see if there are any in vivo inhibitors. So if we have that in vivo inhibitor, we do a clinical study with potent in vivo inhibitor. So presence of significant interaction, yes, study inducers if available. If no, we need to reevaluate what other transporters could be involved. So this is a guideline for a sponsor to navigate through to get an understanding on the DMPK of the compound. So again, FDA provides a detailed decision tree when an investigational drug is a substrate for PGP or BCRP. For example, when we do in vitro bidirectional transportase, say using CACO2 cells or MDR1 overexpressing cells. So if the net efflux ratio is greater than 2, then it could be a potential PGP substrate. So then we need to see if this efflux can be inhibited by one or more PGP inhibitors. So the ratio, the reduction should be more than 50% or to unity if we are administering with this inhibitors. So yes, then it could be probably a PGP substrate, and a complete assessment of non-clinical and clinical information to determine whether an in vivo DDI study is required is something which needs to be understood. For example, in some cases, the drug could be a substrate for both 3A4 and PGP. So then an inhibitor of both 3A4 and PGP needs to be considered. So no, if we get the net efflux, if the efflux is not inhibited by PGP inhibitors, then there could be other efflux transporters like BCRP or MRP2 that could be involved. So so we need to screen if those are the transporters which are involved in the absorption and the clearance of the compound. So that's how the decision tree is laid out. If the net efflux ratio is less than 2, then we need not worry. It is either it's not a poor, it's a poor or a non-PGP substrate. And then we have a decision tree to determine if an investigational drug is a substrate for OATP 1B1 or 1B3. So for example, if your compound has active hepatocyte uptake, and if the drug physiological properties, for example, it has low passive membrane permeability, high hepatic concentrations relative to other tissues, which can be determined in preclinical species, and if it's an organic anion, charged at physiological pH, it could be a potential compound for OATP 1B uptake. So then we would determine the uptake in the expressing cells, which are OATP 1B1 or 1B3 overexpressing cells. And we do compare these values to that in the empty vector. So if the value is greater than 2, and then we would consider that it could be a substrate for OATP 1B OATP substrate. Then now, if your compound is an OATP substrate, then in vivo drug interaction study with a single dose rifampicin or cyclosporine as perpetrator is desired. So that kind of helps you to identify how much of this 1B1 or 1B3 is actually contributing to the disposition in vivo. So also you can alternatively do a PK study in the genotypes of OATP 1B1, which can help the importance of understanding this pathway of 1B1, which could be important for the disposition of the compound. No, then it's not a substrate for OATPs. Similarly, same decision tree is laid out for OCT2, OAT1, and OAT3 expressing cells, OAT3 transporters, where we compare the uptake ratio value to that of empty vector cells. So if the value is greater than 2, then we do recommend clinical study where DDI study with cimetidine for OCT, for example, and probenecide for OET1 and 3 as perpetrators. So that kind of helps you to understand how much this OCT2 or OET1 or 3 is playing a role in the clearance, renal clearance mechanism. If it's not, so then it's not a substrate for OCT2 or OET1 or OET3. So now Suvarchala would be discussing regulatory requirements of transporter evaluation for inhibition studies.
Okay, yeah, thanks, Revathi. So in this slide, like earlier in the slides, we have seen that FDA provided some guidance to the sponsor, especially in the early developmental phase, whether the sponsor should consider evaluating the DDI for certain transporters. Similarly, other regulatory agencies like NMPA, PMTA, and EMA has provided some guidelines to the sponsor to consider evaluating the DDI of the investigational drugs for these transporters, PGP, BCRP, uptake transporters, OATP 1B1, 1B3, renal transporters, OAT1 and 3, and then OCT2 and MATE 1 and 2K. The one exception that we do see for EMA is that EMA does provide a guidance for the bile salt export protein BSEP. So the EMA recommends to conduct a study or evaluate the DDI potential for the BSEP transporters and also the OCT transporter, which the other regulatory agents does not recommend for any study. And then, so similar to the decision tree that we have seen for, to investigate whether the investigational drug is a substrate of certain transporters, similarly here we can see a decision tree for to determine whether an investigational drug is an inhibitor of PGP, OATEPs, and OATs. Starting with the decision tree for PGP, the sponsor should conduct studies to determine if an investigational drug inhibits the efflux ratio or net flux of a known PGP or a BCBRP substrate in CACO2 cells or MDR1. MDR1 overexpressing cells or inhibits uptake of transport or of a substrate when membrane vesicles are used. And to determine the efflux ratio or the net flux, the sponsor need to determine the drug's inhibition potency, that is Ki or IC50 values, and the investigational drug considered to have the potential to inhibit a PGP or BCRP in vivo if the investigational drug, if the IC50, I1 over IC50 or a Ki value is greater than or equal to zero point 0.1 when the drug is administered through parental route, or I2 or over IC50 or the Ki is greater than or equal to 10 when the drug is administered through oral route. Here, I1 represents the mean steady state total peak plasma concentrations following administration of the highest proposed clinical dose, and I2 is the dose of dose of inhibitor in moles over 250 ml if IC50 is in molar units. And if these conditions are not met, like the I1 over IC50 or Ki less than zero point 0.1 and I2 over IC50 or Ki is less than 10, then the in vivo drug interaction study with the sensitive PGP substrate may not be needed. But if the conditions are met, then in vivo drug interaction study with the PGP substrate such as digoxin is recommended. And if these conditions are not met, like the ratios are not less than or equal to zero point 0.1 and less than 10, then the in vivo interaction study is not recommended. Again, if the net efflux ratio or the net flux for the probe substrate is not affected, then the investigational drug is considered either as a poor inhibitor or a non-inhibitor of PGP. So, and then in this slide, what we see is the decision tree to determine if an investigational drug is an inhibitor of OATPs, OATP 1B1 or 1B3. So in this decision tree, what we can see is the sponsor should conduct studies to determine the inhibition potency, that is IC50 or Ki or of the investigational drug on the uptake of a known OATP or 1B3 substrate in cells overexpressing the relevant transporter. And if the Cmax over IC50 of the investigational drug is greater than or equal to zero point 0.1, then the sponsor has to investigate the potential to inhibit OATP 1B1 or 1B3. And to do so, if the investigational drug has the potential to inhibit OATPs 1B1 and 1B3 in vivo, the R value should be greater than or equal to 1.5. And to extrapolate or to estimate the R value, here is the equation to do so, where R value is equals to 1 plus FU times I in max over IC50, where FU or FUP is the fraction unbound in plasma, and IC50 is the half maximal inhibitory concentration, and IN max is the estimated maximum plasma inhibitor concentration at the liver inlet. And this IN max or the maximal inhibitory concentration at the liver inlet is calculated using this equation, where IN max is equals to C max plus Ka times dose times Fa times Fg over QH. And here C max is the maximum systemic plasma concentration of an inhibitor. Um, Ka is the absorption rate constant, Fa is fraction absorbed, and Fg is the intestinal availability, and QH is the hepatic blood flow. And dose is the dose of the inhibitor. And there are certain conditions where you don't know the Fa, Fg, and Ka of the molecule. And in the worst case scenarios, we need to assume that Fa times Fg is equals to 1 and the absorption rate constant equals to 0.1 minute inverse. And considering these uncertainties in the protein binding measurements and the protein binding measurements, the FUP value should be set to 1% if experimentally it is determined to be less than 1%. And if the in vitro studies indicate that it's an inhibitor of OATP1B1 or 1B3, in the sense if the R value is greater than or equal to 1.25, then the sponsor should consider whether to conduct an in vivo study based on whether the likely concomitant medications used in the indicated patient populations are known OATP1B1 or 1B3 substrates. And if the Cmax over IC50 ratio for the investigational drug is less than 0.1, then the in vivo study may not be needed. Similarly, here we see the decision tree to determine if the investigational drug is an inhibitor of OCT2, OAT1, or 3. And in this decision tree, what we can see is a sponsor should conduct studies to determine the inhibition potency, that is IC50 or KIs, of the investigational drug on the uptake of a known substrate for renal transporters in cells overexpressing these transporters. And the investigational drug has the potential to inhibit these transporters in vivo if the Imax or the Cmax over IC50 value is greater than or equal to 0.1. If the ratio is greater than or equal to 0.1, then the in vivo study with the sensitive substrate is needed. If the ratio is less than 0.1, then the in vivo study is not needed. And if the in vitro studies indicate that the drug is an inhibitor of these renal transporters, the sponsor should consider whether to conduct an in vivo study based on the likely concomitant medications used in the intended patient populations and which are known to be a substrates of these renal transporters. So in the earlier slides, what we have seen is a decision to treat to determine if the investigational drug is a inhibitor of different transporters. And when we look at different regulatory guidances, we see a lot of emphasis on the inhibition mechanisms rather than the induction mechanisms. And the reason is that certain transporters like PGP are induced through mechanisms similar to those of the CYP enzymes, like activation of the specific nuclear transporters like PXR and so on. And because of these similarities, information from CYP3A induction studies can inform about the PGP induction studies, and also the in vitro methods to evaluate the induction of PGP and other transporters are not well established. So we don't see a lot of emphasis on the induction of the transporters in most of the regulatory guidances. Then in this slide, we see is the analysis and interpretation of the transporter inhibition assay. So because of the increased awareness about the transporter-mediated DDIs, the regulatory agencies have now made an equal emphasis on evaluating the transporter-based interactions as that of the enzyme-mediated interactions. And eventually, these recommendations have become a part of labeling or summary of the product characteristics. Earlier in the slides, we have seen the decision criteria for, as per the FDA guidelines, for the investigational drug to be considered as an inhibitor of PGP, BCRP, OATP, 1B1, 1B3, and the renal transporters. So similar guidelines have been proposed by NMPA and PMDA for efflux transporters, hepatic uptake, and the renal transporters. And according to EMA, the investigational drug is considered as an inhibitor of PGP and BCRP if the Ki is less than or equal to 0.1 times IGUT, where IGUT is the dose of an inhibitor in molars in over 250 ml. Similarly, the EMA has recommended some guidelines for the investigational drug to be considered as an hepatic uptake transporter inhibitor when the Ki is less than or equal to 25 times Imax unbound inlet, where Imax unbound inlet is the unbound hepatic inlet concentration. And if the Ki is less than or equal to 50 times Cmax unbound, then the investigational drug is considered to be an inhibitor of the renal transporters OAT1, 3, OCT2, MATE1 and 2K as per the EMEA guidelines. And it is evident from the guidelines that for evaluation of transporter-mediated interactions, even though limits or acceptance criteria in terms of inhibition potential were provided for various transporters, a sound scientific justification may also be acceptable, uh, uh, acceptable by these regulatory agencies. So almost all the guidances talk about the utility of PBPK modeling to support and evaluate these transporter mediated DDIs, and application of these PBPK models to guide the clinical studies or designs to support the labeling and waiving of in vivo interaction studies in case of change of dose formulation, subpopulation evaluation, along with the requirements of model verification and limitations of the established models for these TDIs and transporter mediated DDIs or enzyme transporter interplay. And so this slide is basically summarizing this PBPK modeling considerations from different regulatory agencies. And in this slide, the use of various modeling approaches such as static and dynamic PBPK modeling approaches to simulate the transporter mediated disposition, and also to quantitate the transporter enzyme interplay are being increasingly utilized. And this is evident from the figure which depicts the increasing trend in the number of publications over the last decade from 2010 to 2022, where you can see in 2010 there is only one publication considering this transported mediated disposition and transporter mediated DDIs, and the number has increased to 24 by 2022. So we see a significant increase in the number of publications over the last decade, where in silico modeling has been applied to mechanistically explain the transporter mediated disposition either alone or with metabolic enzymes. And the PPPK modeling can aid in predicting the DDIs in complex clinical scenarios such as simultaneous inhibition and induction of enzymes and transporters, and complex transporter enzyme interplay. However, uh, this application of PBPK modeling in these complex scenarios is still in its infancy, and other complex scenarios where PBPK modeling can be applied are transporter-mediated DDI's prediction in genetically polymorphic population or population with other comorbidities like hepatic impairment, renal impairment, or a special population where one need to develop a more sound mechanistic and physiological model to simulate the situation. Then the application of various approaches of TDDIs, which is transporter mediated DDIs, is mainly based on the stage of drug development of the investigational drug is currently in. Um, at at the preclinical stage, only in vitro and animal data are present, and hence to assess its safety and efficacy in humans, static models are initially used. And as the drug development progresses, the available data from clinical studies aid in development development or developing a full dynamic PBPK model of the drug, investigational drug. And this model is then verified, validated, and applied to study the effects of various covariates such as drug-drug interactions, genetic polymorphism, disease state, age, and gender on the pharmacokinetics of the investigational drug. And such approaches are routinely used in drug discovery and development. And this slide here presents the summary or glimpse of the latest literature references where such models were applied to using the different softwares, and the highlighted references here are are the studies where GastroPlus was used in the model development. So if you see here the study by Cato et al, the molecule that they have investigated is edoxaban, and they looked at the impact of PGP on the overall absorption. And this drug cefadroxil, they have looked at the impact of peptide-1 transporter on the absorption of this molecule. And then this slide is a continuation of the previous slide, and this slide shows the references of the literature in diseased populations like hepatic impairment, renal impairment, and cancer population. Again, where they have and also the geriatric population, the special population, where the GastroPlus software was used for model development to assess the clinical PK and overall transporter-mediated disposition and transporter-mediated DDIs uh, for different investigational drugs. This slide talks about the transporter-mediated DDIs in regulatory submissions using through two examples. So the first drug was trametinib, which is a perpetrator, a BCRP inhibitor. So previously, the model was developed for this trametinib using GastroPlus. For other application purposes, therefore, minimal work was required when it comes to the assessment of BCRP inhibition through this drug. And this study or the model using GastroPlus was submitted and accepted by EMA. And the next example is axitinib, which is an intestinal transporter. PGP was assessed, and ACAT model was developed using GastroPlus for this drug to simulate the concentrations in different GI segments. And this work or this model was submitted to FDA and was accepted by FDA. Um, so this slide talks about various strategies for validating the transporter-mediated DDI models. So when it comes to model verification and validation, which are essential for any PBPK model, and transporter DDIs are not an exception for this validation and verification of the model. Before validation, the transporter DDI models need to be verified to assess the model components, and equations, model components, and only after the successful verification, these model validation can be commenced. However, this TDDI model validation against the clinical data comes with enormous complexity as compared to the traditional PBPK models due to the presence of multiple enzymes and transporters and the complex interplay of these transporters and enzymes that defines the ADME of the molecule. And this slide talks about those strategies for performing the TDDI model validation using the external data. In general, it is recommended to validate the model with the single or multiple TDDI studies in order to obtain a confidence in the model. And then, as observed with the metabolic enzymes, depending on the transporters, the fraction transported, the FT can vary, and thus multiple TDDI studies with different transporter inhibitor helps validate this phenomena. Further data from single and multiple doses helps to validate the model with respect to the linearity, autoinduction, or autoinhibition behavior of the compound. And another strategy is that the data available from the individuals with different genotypes can help to validate the model in order to demonstrate its sensitivity to capture this transporter abundance and transporter activity in individuals with different genotypes. As the transporter interactions may not always be evident in the plasma concentration profile, data available in urine or the bile concentrations can help test the model's ability to capture the transporters located in the hepatic or renal tissues. And the other case is where multiple transporters are involved in multiple locations. Uh, um, so these multiple transporters in multiple locations are involved in the drug disposition, thereby the keen observations into the basic PK parameters like the Cmax, AUC, and Tmax can help identify the transporter location being impacted. The other strategy is that additionally data available from multiple doses, different formulations or different routes of administration like oral or IV or modified release formulations can help identify or validate the model with regard to the transporter impact on the absorption, the distribution, or on the saturation kinetics. Lastly, data available from this charcoal administration studies can help rule out the possibility of enterohepatic or the transporter involvement at the bile level in the hepatocytes. Overall, it can be understood that the validation of TDDI or the transporter mediated DDIs models is not straightforward. However, when the model is validated with multiple approaches, as we have seen here, it provides a sound mechanistic explanation, thus increasing the confidence in the modeling approach and the regulatory acceptability. There has been a considerable effort in the areas of transporters over the last decade to understand and build a robust IVIVC for the transporters, but still there is lack of some information when it comes to transporter mediated disposition modeling or the transporter mediated DDI models. So we are lacking the information in regards to the in vitro Km and Vmax values, passive diffusion, protein abundance or expression of the transporters in different tissues, localization of the transporters, transporter induction or inhibition parameters, and whether there is a time dependent inhibition of the transporters or not. So unlike the enzymes, there is still a gap in the knowledge or the in vitro data that's available in terms of modeling the transporter mediated disposition or transporter mediated drug-drug interactions. So this slide here shows the basic model development, validation and application of the PBPK models. So the typical model approach or the approaches that we follow when it comes to the PBPK model development is either the bottom up or the top down approaches. So this bottom up approach is a typical approach that is followed and is initially constructed based on the preclinical data during the early drug discovery stage and compound specific parameters like the physicochemical properties, in silico estimation for the KPs, in vitro data for the transporter kinetics or the enzyme kinetics. So these are used to predict the in vivo PK profile in preclinical species and humans. And then once the model was developed, then the verification of the intravenous disposition prediction in the preclinical species, like assessment of the most appropriate KP predictions, and which take into account the method assumptions, assessment of the prediction accuracy, and other physicochemical properties of the compound. Once verified with the intravenous dose, the next step was to verify the oral absorption prediction or the oral absorption parameters in preclinical species over a range of doses to further assess the prediction accuracy. So this bottom-up approach in large provides a mechanistic understanding based on the in vitro data of the transporter of interest. However, this approach heavily depends on the quality of the in vitro data that's generated, as well as the understanding of the IVIVA data translation. So, so what we do is like we always go for fit for purpose modeling approaches. So either we go for the top-down approach or the middle out approach to support the clinical trial decisions by fitting the existing clinical data, which may require minimal in vitro and mechanistic knowledge compared to the bottom-up approach. And there are many examples in the literature that suggest that the bottom-up approach was successful in recovering the clinical transporter-mediated DDIs, especially when it comes to the renal transporters involving OAT1 and 3 and OAT4, whereas the fit for purpose modeling, like mostly either the top down or the middle out approach is needed to capture the in vivo PK and the transporter mediated DDI for intestinal and the liver transporters like the BCRP or OATP 1B1 mediated interactions. So at Simulations Plus, whenever we want to build a PBBK model for any of the substrate or a perpetrator compound, we always have a standard qualification approach, or what we call is the DDI qualification approach for this model building and validation of that model. So we always start with the literature collection that's collated into the spreadsheet. So in the coming up slide, I can show you a brief snapshot of the literature data, how we collect data and how we organize the data to proceed with the model development and validation. Once the literature was collected using the in vitro data and the plasma concentration profiles for the different clinical studies, if the intravenous plasma concentrations are available, then we'll start with the IV doses, or if the IV data is not available, then we straight away start with the oral doses as a part of model building. And once the model was successfully built and then the verification of single and multiple doses will be carried out, uh, a which gives more confidence into our baseline model, which then will be verified for different mechanisms of the DDI. And when I say the mechanisms of the DDI, it can be the mechanisms through enzyme mediated interactions or the transporter mediated interactions in different locations, like at the intestinal level or hepatic level or the renal conditions. Once the model was built, verified, and verification of all mechanisms of the DDI was performed, um, then all the model building process and the model assumptions are documented in a PowerPoint format and the report format, which can be submitted along with the PBPK reports for the investigational drugs to the distributor. And this in most of the cases, especially for applying for the waivers or any of the uh, propagative further investigation into those compounds, then these reports in the Word document format can be submitted to the regulatory agencies. And when it comes to the assessment of the drug-drug interaction predictions, the current assessment of the DDI prediction success is based on whether the predictions fall within the twofold range of the observed data. And this strategy results in the potential bias towards successful prediction, especially at the lower interaction levels. Uh, so this scenario can bias any assessment of different DDI prediction algorithms if the study contains a large proportion of interactions in the lower range. Therefore, the study conducted by guest Eleanor and his group proposes an alternative and more logical method to assess the prediction success with the variable prediction margin depending on the particular AUC or the Cmax ratios. So the method is applicable. This method is applicable for assessment of both induction and inhibition related algorithms. And the study also addressed the importance of prediction accuracy and performance in the region below twofold change in Cmax and AUC from the regulatory point of view. So to summarize this prediction performance um, or the method, the predictive performance, that is the fold of deviation, is related to the magnitude of the DDI predictions. And the magnitude of DDI predictions, DDI interaction in this study is depicted by R, which is the ratio of the observed post and pre-DDI values, which can be for Cmax or AUC. And if this magnitude of the DDI predictions are the R value are greater than the acceptable limits for the in silico DDI predictive performances are wider. So if we see here, if the DDI interaction ratio, which is the Cmax or AUC ratio, is equals to 1, we see that the value is close to unity. And once the um, uh, once the ratio of the observed post and pre-DDI values are greater than 1, we can see this line pull off becoming wider compared to the unity line. So, so until now, what we have seen is the basic assumptions or the model considerations, the challenges, and how we approach when we are building a DDI, a PBPK model for any DDI standards. So now we will look into one of the case study or the case or a PGP substrate digoxin, where we built the physiologically based pharmacokinetic model and then validated for known drug-drug interactions with the PGP inducers and inhibitors like rifampicin and itraconazole. So as we have seen in the earlier slide, where we have presented this DDI qualification approach, we always start with the literature collection. So here in this slide, what you can see is the snapshot of the spreadsheet that we have created as a part of literature collection for the PGP substrate digoxin. So we start with the physicochemical properties, and then we collect the relevant data in terms of solubility, metabolism, if any, transporter processes, permeability, and so on. And also we collect the clinical data for both IV and the oral doses to build and validate the baseline model. And then the DDA interaction studies with the multiple perpetrators, PGP inducers and inhibitors, um, for single day and multi-day administrations. So we always start with developing a modeling strategy to build a PBPK model. So the first step is to collect the physicochemical and the biopharmaceutical properties to build the baseline model. And once the baseline model was developed, then we'll validate this model or verify this model in the IV dose if the data on after IV administration is available. And then and also a single oral dose in healthy volunteers. Once the model does perform reasonably in predicting the plasma concentrations after an IV and oral administration, then the further verification with the multiple doses and the single doses in different conditions like the fasted and the fed state. In some cases, we can test if the data is available. We can test the model in different ethnic populations like Caucasians, Japanese, and also in different phenotypes or the genotype individuals. Once the model verification was completed, then sorry, model building and validation was completed, then the next step was to verify all the mechanisms for DDIs. Here the digoxin is a substrate for the PGP, so the DDI interaction that we are validating here is the inhibition of PGP through using rifampicin and itraconazole, which are PGP inducers and inhibitors. So this slide here is kind of summarizing all the input or the key physicochemical and the biopharmaceutical biopharmaceutical parameters for digoxin that were used in the GastroPlus simulations. So looking into the digoxin characteristic properties, this digoxin is a prominent victim drug to investigate the PGP-mediated DDIs, as its disposition is mainly governed by PGP, and the primary route of digoxin elimination is through renal excretion via glomerular filtration and tubular secretion of the unchanged drug. And the studies also indicate that digoxin is a substrate for an OATP uptake, and this study was conducted in the rat small intestine. And then digoxin is also a substrate, or it acts as a sodium potassium ATPase acts as a receptor for digoxin, and this sodium pump in skeletal muscle constitutes the main determinant for volume of distribution, and tissue binding is the rate limiting step for in the digoxin distribution kinetics. Digoxin is a hydrophilic drug which exhibits low passive transcellular flux, and it is metabolically stable in humans, and its absorption, distribution, elimination is mainly through transporter-mediated process. Um, the PGP attenuates the intestinal absorption and the central nervous system distribution, and is involved in the elimination by transporting it from hepatocytes to bile and renal proximal tubule to urine. And basolateral uptake of digoxin has been shown to involve the sodium-dependent processes, and digoxin is eliminated as an unchanged drug with a comparable recovery in urine and feces in humans. And when it comes to the metabolism, digoxin is not metabolized to an appreciable extent, although a lactone ring reduction by gut bacteria following oral administration has been observed in some subjects. And because of the absence of CYP-mediated metabolism, which is unique to humans, so this absence of CYP-mediated metabolism is unique to humans and can be extensively found in animals. When it comes to the biliary excretion involves the hepatic uptake via sodium dependent transport process based on the in vitro data, and then the subsequent PGP canalicular excretion into bile. The hepatic uptake of digoxin was believed to be mediated by OATP, but we don't have any data available in the human study, but the study was conducted in rat hepatocytes. When it comes to the renal clearance of digoxin occurs by both glomerular filtration and tubular secretion, where the active secretory component of the renal clearance involves the renal proximal tubules by the sodium dependent process and OATP4C1, followed by secretion into urine via PGP. So this slide basically summarizes all the transporter mediated processes that enables the absorption, distribution, and elimination of digoxin. Further survey into the literature, we found that digoxin is also a substrate for MDR3 PGP protein, where the study conducted by Smith and group reported an increase in the bidirectional transport of several PGP substrates such as digoxin. But to start with, we don't have any in vitro data available like the Km or the Vmax values. So we started with understanding the expression level of this MDR3 PGP transporters in different tissues. And we can see that the expression, this MDR3 PGP transporter is highly expressed in liver with minimal expression in kidney. So we know that this transporter is highly expressed in kidney, and we don't have any in vitro data like the KM and Vmax to start with. So the way that we incorporated this into the model is through the apical PSTC in liver. with the apical PSTC of 0.5 in liver. So with this understanding of the in vitro data, the distribution characteristics, and the other transporters that are involved in the absorption ADME of this digoxin, we built the basic baseline model for this digoxin and then further verified in multiple IV and oral administrations after single and multiple doses. And in this slide, what we are seeing is the results of the model validation of digoxin, where we compared the observed Cmax and AUCs to the predicted Cmax and AUCs. And here the purple lines represents the comparison of the observed and the predicted Cmax, and the blue circles represent the comparison of the observed and the predicted AUCs. The red lines represent the twofold prediction errors. The black line represents the 1.25-fold prediction errors, and the green line represents the unity. And when we look at the overall results, around 50% of the predictions are close to the unity line, and 70% of the predictions are within the 1.2-fold criteria, and all the predictions are within the two-fold criteria. So this gives us a confidence into the baseline digoxin model, which can be further used or used for the verification of the different mechanisms of the DDIs. So with that confidence in the model, the next step was to look at the prediction accuracy for the DDIs, and we looked at the interaction potential or the magnitude of the DDI change when digoxin is co-administered with the PGP inducer and inhibitor rifampicin and PGP inhibitor itraconazole. And the green circles represents the AUC and Cmax comparison of the observed and the predicted values for rifampicin, and the orange circle represents the Cmax and AUC comparison for itraconazole. And this red lines represents the twofold prediction errors, and this black line represents the error as per the guest criteria that we have seen in the earlier slide. So when we look at the overall prediction accuracy for both Cmax and AUC when digoxin is co-administered with the PGP perpetrators rifampicin and itraconazole, we can see that the model reasonably predicted both the Cmax and AUC, and most of the predictions are close to the unity line, and if not, they are within the twofold prediction errors. Then we'll move to the next case study or the next example of the PGP substrate fexofenadine. So fexofenadine is a substrate of multiple transporters like PGP, OATP2B1, and OAT3. So, and once we build the model incorporating all this transporter mechanisms, the model that was then validated for known DDIs with rifampicin, itraconazole, verapamil, and efavirenz. And all these four perpetrators are the PGP inducers or inhibitors. So similar to what we have seen for the digoxin, we started with literature collection, compiling all the in vitro data, the physicochemical properties, transporter-mediated processes, polymorphisms, and then the clinical PK studies after IV and oral administration, and then the clinical DDI studies with multiple PGP inducers and inhibitor. Once the data was collected and compiled, then we started with developing a modeling strategy for this fexofenadine baseline model. Uh, so using the biophysicochemical and the biopharmaceutical data, baseline model was developed, and then the model development was carried out against the microdose of oral and IV doses in Caucasian and Japanese healthy volunteers. And then the model was then verified against the multiple doses and the single oral doses in Caucasian and Japanese subjects, both in the fasted and fed state. Once the model was verified against single and multiple doses, then the model verification for DDI mechanisms was carried out with rifampicin, itraconazole, efavirenz, verapamil, single and multiple doses. And the DDI mechanisms that we have investigated here is the induction and inhibition of PGP and inhibition of OATP2B1. And the slide here summarizes the key physicochemical and the biopharmaceutical parameters of fexofenadine that was used in the GastroPlus simulations. So this fexofenadine is moderately lipophilic compound with moderate solubility, and it has low protein bounding. And this fexofenadine is usually administered in the racemic mixture form, the R and S forms, where they exhibit differences in tissue distribution, which is uh, which is resultant from the differences in terms of the protein binding for R and S form. And because fexofenadine is poorly metabolized by CYP enzymes or the isoforms, it its pharmacokinetics primarily dependent on the activities of the PGP, OATP, 2B1 transporter at the gut level, and the renal uptake transporter OAT3. So, as a part of model development for fexofenadine, we have considered muscle and kidney to be permeability limited tissues, and the passive diffusion in all of the tissues is described by, usually the passive diffusion in tissues is described by the PSTC value that's scaled from the liver PSTC according to the individual tissue cell volumes. So to obtain the PSTC that is scaled from the liver PSTC, we have optimized the specific PSTC to 6 e power minus 4 milliliters per second per milliliter. So using all the in vitro data and defining this passive diffusion through optimizing the specific PSTC value, the model was built and then verified after multiple IV and oral administrations in different ethnic populations and both in fasted and fed state. uh, so this slide here presents a model validation of fexofenadine similar to digoxin. What we have seen, we have compared the observed Cmax and AUCs to the predicted Cmax and AUCs, and here the purple circles and the blue circles represents the Cmax and AUCs. Red lines represents a two-fold error lines, black lines represents the 1.2-fold prediction errors, and green line represents the unity. When we look at the overall performance of the baseline model, for most of the studies, the predictions are close to the unity, and at least for 70% of the studies, the predictions are within the BE limits, which is 1.2-fold prediction errors. With this confidence in the baseline model, the next step was to look at the DDI prediction accuracy. For fexofenadine in presence of multiple inducers and inhibitors of PGP. And we can see that for the most of the predictions, the observed Cmaxes compared to predicted Cmax are close to the line of unity, which is this green line. If not, at least they are within the guessed criteria for multiple perpetrators or the inducers or inhibitors of PGP. I think, Revathi, you can take the floor to continue with these case studies.
Sure. Let me see if I can get the control. Okay. Okay. So now we will be discussing about the PBPK models for OATP1B substrates like rosuvastatin, which is also a BCRP substrate. So, and the model validation with DDIs with rifampicin and zemfibrozil. Coming to the schematic view of enzymes and transporters involved in the absorption and disposition of rosuvastatin in gut, liver, and kidney of human body. So this rosuvastatin is kind of minimally metabolized by 2C9 and UGT1A1. So there is a hydroxy metabolite and the glucuronide lactone metabolite essentially formed from 1A1, but the metabolism is less than 10%. So the major drivers for hepatic disposition are OATP 1B1, 1B3, which are the major drivers for the hepatic uptake, but there's also minimal contribution from OATP 2B1 and NTCP. So the way the FT is calculated for each of these transporters is based on an in vitro paper where they estimated the fraction transported by each of these transporters. So and there is also some percent of the compound that is eliminated renally, which is primarily mediated by O3 at the basolateral side and BCRP into the urine. So this is a modeling strategy for rosuvastatin. Here we have built a baseline model using the in silico, in vitro, and clinical PK data, where the model was first initially developed for a single doses, where IV infusion 8 milligram and oral tablet 5, and the 40 milligram was used for model development. Once we felt that these two doses are described well, we further verified for other doses where they are given as a single doses, ranging from 5 to 80 milligrams. So entire dose range is covered. So while we are developing the model, we are also parallelly trying to verify the verifications with the DDIs. Essentially, here we are looking with the DDIs for single dose IV and oral infusion. We are verifying the FT contribution from for OATP 1B1, 1B3, 2B1, NTCP, O3, and BCRP. So, and we also tested the effect of multiple oral dose of rifampicin, essentially inducing some of the enzymes like 2C9 and UGT1A1. And then we also have the DDI study with zemfibrozil, where zemfibrozil is also an inhibitor of OATP 1B1 and 1B3, and other transporters like NTCP and O3. So, and we also have a zemfibrozil which inhibits 2C9 and 1A1, but since the metabolism is less, the majority of the DDI is causing because of the inhibition of these transporters. So here are the physicochemical and the biopharmaceutical properties. So here, most of the values are coming from the literature values. Only the Vmax values of the enzymes and transporters are optimized based on the information that we have with respect to FM and FT, which is where it is coming from a combination of in vitro and in vivo data. So when we look at the model validation of rosuvastatin, here we are looking at the model validation with respect to Cmax and AUC, the PK parameters. So as you can see, when we compare the predicted versus observed values, most of the values are within the 1.25 fold criterion for both Cmax and AUC, and almost all of the values are within the 2-fold criterion. Since this compound, the reason why we had a higher comparatively bigger criteria for this compounds is because this compound is a substrate for OATP 1B1, which is highly polymorphic across the different ethnicities. So we employed a higher model value evaluation criteria for the same reason. And then we also had a DDI model verification. We used the compounds rifampicin and zemfibrozil, which are strong and moderate inhibitors of OATPs. So as you can see, most of the predictions for both Cmax and AUCs are closer to the line of unity, and most of them are within the 1.25 fold criterion. So this explains that whatever the FM and FT contributions we attributed to different enzymes and transporters are very much in line with what clinical data suggests. So that is with rosuvastatin, which is an OATP and a BCRP substrate, and it's recommended by FDA whenever we are investigating the inhibitory potential of new drugs. So we also have pravastatin, which is a OATP 1B substrate, and we'll also discuss the model validation with DDIs with rifampicin, zemfibrozil, fluconazole, and itraconazole. So coming to pravastatin, regarding the enzymes and transporters involved in the disposition of pravastatin in gut, liver, and kidney. So here, when we give oral pravastatin is a substrate for MRP2, which is a gut efflux transporters, prevents the drug from absorbing. And it's a substrate for 3A4, which kind of is present in the gut as well as in the liver, which kind of metabolizes pravastatin, but it's somewhere around 30 to 50% is metabolism coming from these enzyme. But majority of the pravastatin is taken up into the liver by transporters like OATP1B1, 1B3, and it is also it gets into the bile by MRP2, which kind of affluxes the drug into the bile. There's also significant clearance through a renal route, which is primarily mediated by O3 and also the MRP2 or the apical, which kind of gets the drug into the urine. So these are the enzymes and transporters involved in the absorption and dispersion of pravastatin. So here for pravastatin, we don't have the FT contribution from OATP1B1 versus 1B3. So we attributed the entire FT to 1B1. So where there was no sufficient data from in vitro data that kind of explains what is significant, what is the individual contribution of each of these transporters. But whereas for rosuvastatin that we looked at in the earlier slides, we do have in vitro data that kind of suggests what is the contribution from 1B1 versus 1B3. So that's the assumption we made. So just to let you know that whenever we are modeling the statins, we kind of assume the liver and kidney as a permeability limited tissue so that we can incorporate this transporters and the passive diffusion into these tissues. So again, this is the modeling strategy we employed. Again, it involves the construction of baseline model based on physicochemical, in silico, in vitro, and clinical data. So the model was initially developed for IV infusion and oral solution, and then once we had confidence on the the PK data that was in accordance with the observed data, then we kind of try to see other doses ranging from 5 to 80 milligram, which is the entire dose range of pravastatin, and also the multiple doses and DDI studies. So this is a parallel work that we do to verify the DDIs with our models, where we employed rifampicin, which is an inhibitor of OAT1B1, 3A4 and O3, zemfibrozil, which is an inhibitor of OAT1B1, O3, and itraconazole and fluconazole, which are inhibitors of 3A4. So here what we are trying to do is here we are verifying the contribution of OATP 1B1 and O3s using these transporters, using the inhibition constants that have been nicely validated with other compounds. So the interaction potential will remain the same, but here we are testing the effect on pravastatin. So similarly for itraconazole and fluconazole, the inhibitory potential, for example, the Ki and IC50 values have been previously verified with CYP3A4 substrates like midazolam, alphentanil, and triazolam. And so we use the same inhibition constants to see how much, if we can reproduce the DDI with the pravastatin. So it's a continuous validation process that we do across the compounds. So here are the again the physicochemical and biopharmaceutical and ADME processes that we incorporated. So most of the values are from the literature, except the Vmax values, which are often optimized to see if we can reproduce the clinical DDIs, and also if any information is available with respect to FM and FT from a combination of in vitro and in vivo data. So as you look at the data here, again, we do have the predicted versus observed data for Cmax and AUC. As you can see, most of the data are within the twofold criteria. Again, we employed the twofold criteria here because of the polymorphisms in OATP1B1, which kind of gives a wide range of values across clinical studies. And then we also have DDI studies with zemfibrozil, rifampicin, itraconazole, and fluconazole. As you can see, the DDIs are mostly within the 1.25-fold criterion, but all of them within the twofold criterion. So this kind of reaffirms that the FMs and FTs that we have attributed for the pravastatin absorption and disposition are more or in line with what we see in the in vivo. So, okay, so we have discussed that we have developed models of PGP substrates and OATP1B substrates, and we also have models which are validated, which are OATP1B1 inhibitors like rifampicin and zemfibrozil. So what do we do with these models? How do we apply for regulatory submissions? So for example, if your investigational drug is a victim drug, so if you think that your compound is a victim for any of these transporters, so we do have the in vitro metabolism data and the transporter data from the in vitro, and most likely based on whether which mechanism is prominent, whether the metabolism or transporter pathways, the FDA recommends doing a clinical DDI with strong inhibitor like rifampicin or a mass balance studies to get an initial estimate of FMs and FTs by each of these pathways. So once we know the relative contribution of enzymes and transporters, what we can do is we can test the effect of inhibitors. For example, here they have done we have done a clinical study with a strong inhibitor like rifampicin. Now we can do a scenario where we use a zemfibrozil model, which is previously verified with substrates like pravastatin, rosuvastatin, or atorvastatin. So we can test the effect of zemfibrozil on the current investigational drug. So we can employ to produce a scenario where how much the effect of zemfibrozil could be on the investigational drug. Similarly, even if a client didn't do a clinical study with rifampicin, we can use our rifampicin model to see how much the possible impact could be. But more often times for that, we need a substantial mass balance study that could support the FM and FT contributions for the investigational drug. So you might be wondering what are the interaction parameters, that is inhibition and induction parameters that we optimized or obtained from the literature for the perpetrator models that we developed. For example, rifampicin or zemfibrozil and its glucuronide, or itraconazole and fluconazole. So these interaction parameters are listed in towards the end of the presentation, which are well verified using substrates for OATP1B or PGP or BCRP. So, coming to the case scenario where the investigational drugs are transporter inhibitors, for example, transporters like OATP1B or PGP or BCRP. So, using the in vitro Ki value that is generated, if the R values calculated are higher than the cutoff values that Suvarchala has discussed in the earlier slides, then we can use a relevant substrate, that is, our compounds in the DDI matrix can be used. For example, you can test the effect of investigational drug if it is an OATP1B inhibitor using our victim compounds like rosuvastatin and pravastatin, and see how much this investigational drug affects the PK of the victim, like rosuvastatin and pravastatin. So depending upon the predicted magnitude of interaction, whether it is significant or not, in the former case, a clinical DDI study is still required, whereas in the later, a sensitivity analysis will suffice. So in this way, we are going to test the effect of investigational drugs if they have this inhibitory potential towards the any transporter. So this way we kind of use our PBPK models to test the scenarios where when the compounds are when the compounds have some inhibitory potential towards transporters. Okay, next is like in terms of additional literature on GastroPlus DDI applications, here are some of the publications that you can refer, how the DDIs can be used, which kind of gives you a workflow and direction to follow which enzymes and to what kind of data is required, how do we develop a modeling strategy, and how do we further verify DDIs. So you can refer to these papers. Alternatively, you can request for the PBPK reports of the models that we developed in-house that can give you a sense of how model is developed and verified for victims or the perpetrators. So this is another set of examples. So we also have another important resources with respect to guidances from different regulatory agencies, whether it be FDA, EMA, PMDA, or ICH. They provide a thorough guidance on following the steps when a new drug is being developed to help navigate, understand the DMPK of the compound. So with this, we end our presentation and we thank you for your time and we'll look forward to the Q&A session.
All right, before we start answering some of the questions I see here, I have a second poll that I will be launching. And I'll give everybody a couple seconds there to answer those, and then we'll jump into the Q&A. And this last poll has a total of 4 questions. So make sure to answer each one. I'll give everybody a couple more seconds here. Okay. Then I'll go ahead and end it there. Okay, so I do see a couple of questions here. We'll start with the first one. It says the turnover rate of all enzymes and transporters is set to a fixed value. How significant is the turnover rate in interaction?
So hi Ami. Thank you for that question. So yeah, in GastroPlus we do have the turnover rates set to a particular value. This is because these values were established way long back when we don't have much of the information with respect to turnover rates. But now that since we have a better consensus with the turnover rates, since even ICH has published turnover rates in of different enzymes in its guideline. So we are looking into that. But for making any DDI predictions, it shouldn't matter because you're starting from a single baseline value and you are accordingly optimizing your Vmaxes or clearances to get a baseline, and then you're predicting the DDI with your constant. So it shouldn't affect, but yeah, there's literature coming with respect to different turnover rates. Yeah, thank you for the question.
Okay, we have a couple more. You're also asked if the turnover rate of all enzymes and transport. Oh, we answered that one. Sorry. Is the post-console model as perpetrator published, or can the report be shared?
Posaconazole, I'm not yet sure. I need to check with one of our chief scientist. But I think we do have a model, a baseline model. I'm not sure if it is verified for DDIs, but Vaibhav, you can reach out to us. You can write to our support team to see if you wanted to have an access to that model.
Awesome, thank you. I have another one here. It says, could you please elaborate how you address the uncertainty in vitro KI for DDI issues with selectivity?
Yes. There is a lot of uncertainty in the KI, especially that is determined across different laboratories. But since ultimately you have to have some clinical DDI study to see what KI is working. Especially when it comes to new compounds and they have certain KI value. So regulatory agencies recommend doing a sensitivity analysis going tenfold lower to see how much profound impact it could have. But yeah, there is still this uncertainty. But with more and more data that is coming with the clinical data, I think those KIs can be verified and established across different compounds, like across different 3FO substrates or different OATP substrates. So it's a kind of a retrospective analysis as well.
We have one more, and it says, how does biomarker data, how will it be helpful in achieving confidence, and how do you use it to inform PBPK models?
Oh, yeah. So, yeah, there is a lot of biomarker data that is coming, especially with respect to OATPs and OATs, and especially coporphyrin, where they are trying to estimate how much of the coporphyrin is changing with respect to any interacting compounds. So it is being evolving. The literature is coming up. I think it would be most useful. I think some have recently started building PBPK models for coporphins, which can give which can help us understand how much was the DDI effect could be. Yeah, this is a part of literature that is evolving in the recent times.
Okay, and then I think we're going to do one more. This was originally a seed question, and it says, what are the main applications of PBPK models submitted to regulatory agencies such as the FDA for evaluating transporter-mediated DDI, and which applications are most widely accepted by regulatory agencies?
Oh, so do you want to take it, Suvarchala?
Yes, Revathi, I can take it. So when it comes to like there is one of the review by US FDA's office from 2008 to 2017, which kind of suggests that majority of the applications are DDI based, whether mostly the enzyme based, and very small fraction are transporter based applications. And when I say the DDI, most of the applications are a combination of enzyme and transporter based application. So overall, when we look at the number, DDIs are the major, contribute to the major applications to the regulatory agencies, followed by the pediatric assessments, uh, renal and hepatic impairments, food effects, and then very small portion like 2% or pharmacogenetics. And if there are no more questions, I think of one of the question by Aditya is that he wants to know how we can enter or how we can tell the program how often the perpetrator can be administered. So maybe I can pull up the slides and quickly show that. So yeah, so Aditya, here you can see in the DDI module under the perpetrator, you have this dosing information here, and you can specify or tell the program what could be the dosing interval of the perpetrator, how often the perpetrator can be administered. Like this is where you can do that if you don't want to create an MTD file in the PPPK model. And similarly for the substrate, uh, if you don't have an MDD file, here you will have what's the substrate dose and what is the dosing interval, like how after how long the perpetrator the victim drug was given. So that's kind of information you can enter here if you don't have an MDD file. Hope that helps and answers your question.
Thank you, Suvarchala. I think I'm going to do one more. And then we can wrap it up. So the other one was how to characterize the role of transporters in the ADME process of drugs more accurately and reasonably. For example, what in vivo or in vitro observational data can accurately reflect the specific contribution of a transporter?
Yeah, so first to characterize the role of transporters, we need to make sure whether the compound is substrate of any of the influx or the influx transporters. And that brings us back to the decision tree that we have seen in the slides earlier. So based on the decision tree, we need to first understand whether it is a substrate of PGP, OATPs, or OATs. And once we know whether it is a substrate or of an efflux and influx transporter, then we need to understand the common PK parameters, like the parameters that would affect your the plasma profile, like the permeability, solubility, because it all revolves around the BCS classification. Like if the compound is a class 1 compound with a high solubility and high permeability, it is well absorbed across the GI tract, so the effect of transporters would be very minimal. And for the BCS class 2 compounds, because of the low solubility, the gut transporters could play a significant role on the overall disposition of the compound and so on. So we need to have an understanding of the permeability, the solubility, the BCS classification of the compound to characterize or to more understand the role of transporters in the ADME process of the drugs.
And I just see one that came in the chat and it says, may I know if it is possible to evaluate a DDI with a biologic inhibitor and victim in terms of transporter?
Revathi, do you want to take that? Because I'm not sure if we can do that with biologics.
Yes, I think with monoclonal antibodies, there are compounds, also ADCs, where sometimes your compound, the drug you're giving, could interact. So we could do that in our GastroPlus. Yes.
Awesome. I'll wrap it up. I want to say thank you to everyone. For any questions you still may have, you can always reach out to our business development team or our scientists, and we can follow up with answers. We invite you to learn more about DDIs on our website. I put a couple links in the chat. You can also follow us on our social medias. And then you or your colleagues may also be interested in our upcoming webinars on GPX. And then you can find links to that on our website as well. This webinar has been recorded for playback and will be available on our website and YouTube channel. This concludes our webinar for today. Thank you, everyone.